In this episode of Toxicity Tests, we're going to look at endocrine effects stroke endocrine disruptors. Firstly, let's define what we mean by endocrine disruptors. This slide is taken from the AllTox website. Its title is Community Strategy for Endocrine Disruptors. By community it means the European community. It reads, endocrine disruption refers to adverse effects on reproduction, development, or health resulting from chemically induced interactions with the body's hormone system. A range of synthetic as well as naturally occurring agents have been identified as potential endocrine disruptors, including pesticides, pharmaceuticals, plastics, and other chemicals. This slide, taken from Canadian Council on Animal Care, shows the conventional test method in the left-hand column. It reads, Two-Generation Reproductive Toxicity, OECD TG416, and that is the test we're going to look at now. OECD test 416 is an animal test. It involves testing substances on rats for 2 generations to see what effects these chemicals or other substances will have on the rats. This slide concerns itself with the initial considerations of the test. It reads, this guideline for 2-generation reproductive testing is designed to provide general information concerning the effects of a test substance on the integrity and performance of the male and female reproductive systems, including gonadal function, estrous cycle, mating behavior, conception, gestation, parturition, which is about childbirth, lactation and weaning, and the growth and development of the offspring. The study may also provide information about the effects of the test substance on neonatal morbidity, mortality, and preliminary data on prenatal and postnatal developmental toxicity, and serve as a guide for subsequent tests. So that is what the initial considerations are for this test. This slide concerns itself with administration of doses. It reads, the animal should be dosed with the test substance on a 7 days a week basis. The oral route of administration, diet, drinking water or gavage, is preferred. If another route of administration is used, justification shall be provided and appropriate modifications may be necessary. All animals shall be dosed by the same method during the appropriate experimental period. When the test substance is administered by gavage, this should be done using a stomach tube. So that is how this test is undertaken. This slide explains to the scientists what to do after all the animals have been killed. It reads, gross necrophagy. At the time of termination or death during the study, all parental animals, all pups with external abnormalities or clinical signs, as well as at least one randomly selected pup sex litter from both the F1 and F2 generation shall be examined macroscopically for any structural abnormalities or pathological changes. Special attention should be paid to the organs of the reproductive system. Pups that are humanely killed in a moribund condition, that means a dying condition, and dead pups, when not macerated, which means they're not sodden, should be examined for possible defects and or cause of death and preserved. This slide concerns itself with the evaluation of the results. It reads, the findings of this 2-generation reproductive toxicity study should be evaluated in terms of the observed effects including necrophagy and microscopic findings. The evaluation will include the relationship or lack of between the dose of the test substance and the presence or absence, incidence and severity of abnormalities including gross lesions, which are injuries, identified target organs, affected fertility, clinical abnormalities, affected reproductive and litter performance, body weight changes, effects on mortality and any other toxic effects. The physico-chemical properties of the test substance and, when available, toxicokinetics data should be taken into consideration when evaluating test results. At the bottom it reads, a properly conducted reproductive toxicity test should provide a satisfactory estimation of a no effects level and an understanding of adverse effects on reproduction, parturition, lactation, postnatal development, including growth and sexual development. This is the last slide from the OECD test 416. Interpretation of results. A 2-generation reproductive toxicity study will provide information on the effects of repeated exposure to a substance during all phases of the reproductive cycle. In particular, the study provides information on the reproductive parameters and on development, growth and survival of offspring. The results of the study should be interpreted in conjunction with the findings of subchronic, prenatal, developmental and toxicokinetic and other available studies. The results of this study can often be used in assessing the need for further testing of a chemical. Extrapolation of the results of the study to man is valid to a limited degree only. This isn't the first time we've seen extrapolation of the results of the study is valid to a limited degree. It's been on other tests, and it is possibly a reason why there are quite a few other alternative tests here that are either reduction tests or replacement tests, but there still doesn't seem to be that will to completely reject the animal tests and go for total alternative tests. In fact, I don't know of any of the tests that I've come across yet that have had alternative tests, non-animal tests, as a total replacement for the animal tests. But we live in hope. Anyway, now we're going to look at some of the alternative tests for endocrine disruptors. This first slide we looked at earlier. As we can see, the conventional test is in the left-hand column, and the second column, it says name and description. That is the first alternative test we're going to look at, and it's called microsomal aromatase assays. So let's have a look at that now. The first thing to say about the microsomal aromatase assay is it is used in conjunction with either pubertal male in vivo assay or adult male in vivo assay. So it looks like it's used in conjunction with a animal test, so that's not too good. But anyway, it says a human placental microsomal aromatase assay and the human recombinant microsomal aromatase assay are in vitro assays for measuring aromatase and aromatase inhibition. Aromatase is the cytochrome P450 enzyme complex responsible for the conversion of androgens to estrogens during steroidogenesis. Alterations in the amount of aromatase present at its catalytic activity will alter the levels of estrogens in tissues and disrupt estrogen hormone action. Inhibition of aromatase alters the catalytic activity of the enzyme and results in a rapid decrease in the levels of estrogens. It says the validation status. It's validated by, the Environmental Protection Agency for identifying aromatase inhibiting chemicals. This alternative test is called in vitro androgen receptor binding assay, and in brackets it says rat ventral prostate cytosol. So this sounds like it's not a totally non-animal test, because I imagine they're using the cells from a rat to use in the in vitro test. So although the rat is not going to be experimented on live, as is the case in a lot of the tests obviously, it still loses its life in order that this test can take place. In the fifth column, under reduction, it says a negative result in the Tier 1 test battery is sufficient to classify a chemical as having low or no potential to cause endocrine disruption, thus avoiding Tier 2 animal tests. So, in that way, it's a good test because at least it does reduce some animal suffering. Okay, let's have a look at the next test now. Okay, this test is called the Human Recombinant Estrogen Receptor Binding Assays. And the next slide will tell us exactly what this one is about. The description for this test reads, these assays identify the potential of the test compound to interact with and disrupt estrogen systems. The test compound is measured for its ability to bind to estrogen receptors, ERs, as well as its affinity for the ERs in comparison to a standardized estrogenic compound. Under validation status, it reads the OECD is presently conducting validation studies. In the US in 2003, it was recommended that recombinant estrogen receptor binding assays undergo validation studies, though they still require standardization. Under regulatory status, it says OECD, draft of a new test guidelines proposed. So it does look like this will be accepted by the regulatory authorities, this test. However, it seems to be only accepted for Tier 1 screening, so it's not going to replace the animals, unfortunately, but it might reduce animal usage due to the fact if it doesn't look like they are endocrine disruptors in these alternative tests, then it will not go on to animal tests. That's my understanding anyway. Okay, let's have a look at the next one now. Here are two more alternative tests to the animal tests. I'm just going to look at the top one, which reads Stably Transfected Human Estrogen Transcriptional Activation Assay for the Detection of Estrogenic Agonist Activity of Chemicals. Crikey. Okay, let's have a look, see what that says. The description of this test reads the HER-HELA-9903 cell line is used to detect transcriptional activation mediated through human estrogen receptor alpha. When estrogen binds to human estrogen receptor alpha, the receptor ligand complex is translocated to the nucleus where it activates a luciferase reporter gene. Bioluminescence is measured with a luminometer. Okay, so that's how that test is undertaken. The validation status reads OECD presently conducting validation, and the lead agency is the Interagency Coordinating Committee for the Validation Alternative Methods, which is the American body for the alternative methods. Under regulatory status, OECD draft new TG 455 2008. So that means it's pretty much got full regulatory status, this test, to be included, in an OECD draft. I suppose it only says draft, but I think it has been accepted as a full test now. What does it say at the bottom? It says reduction. A negative result in the Tier 1 test battery is sufficient to classify a chemical as having low or no potential to cause endocrine disruption, thus avoiding Tier 2 animal tests. We were talking about that earlier. Okay, I'm just going to actually show you this OECD test to show that it has got very high status, this test, and it is accepted by the regulatory authorities, but of course it's not a full replacement. So here it is, the OECD test 455, stably transfected human estrogen receptor alpha transcriptional activation assay for the detection of estrogenic agonist activity of chemicals, and it was adopted on the 7th of September 2009. I'm not going to look at the actual test, but one thing I would say about the alternative tests are you're going to need a much more skilled workforce to do them. This is one drawback. It doesn't really take much education to do the tests using the animals, but you're going to have much more skilled scientists to do these tests. And I wonder if that's one of the reasons why there seems to be hesitation in introducing them in a more comprehensive way. This is another test. This is a penultimate test, so you can see how many tests, alternative tests there are to detect endocrine disruptors. This one's called H295R cell-based steroidogenesis assay, and it says the OECD is presently conducting validation. The lead agency is the US Environmental Protection Agency. Under reduction, again, it says a negative result in Tier 1 test battery is sufficient to classify a chemical as having low or no potential to cause endocrine disruption, thus avoiding Tier 2 animal tests. Okay, we're going to look at the last test now. This last test by Certichem, which I imagine is a company, is called the MCF-7 cell proliferation assay for estrogenic activity. And it says an intra-laboratory validation study has been initiated at Certichem Laboratories. ICCVAM has recommended that the assay be given a high priority for additional validation. So they'd only do that if it looks like it had very good potential to help in the identification of endocrine disrupting chemicals. Anyway, let's have a little bit more of a look at this test before we end this episode. The description of this test reads: this measures whether and to what extent a substance induces cell proliferation via estrogen receptor mediated pathways. Testing is done using MCF-7 cells, an immortalized human breast endocarcinoma cell line that endogenously expresses ERs, that's estrogen receptors. When it says immortalized human breast, that means cancer cells, unfortunately, that are being used. I mean, obviously there's nothing unethical about using immortalized human breast cancer cells, other than the fact that someone had to have them initially, I suppose. But the thing is about these cells is they just keep growing and growing and growing. They don't die off like other cells. So from that point of view, they're really useful. In fact, I believe that there's cancer cell lines that have been kept alive since the 1950s. Anyway, so in conclusion, well, whether this is scientific or not, whether the animal tests are scientific or not, is debatable. Even by their own admission, they said that the results are only able to be extrapolated to man to a limited degree. So perhaps really we need this battery of alternative tests to be measured against the animal tests, see which one is the best, and if the battery of alternative tests is the best, then this looks like a prime candidate for full replacement, and thus the end of using animals to test for endocrine disruptors.